Hello friends, in the last class we have discussed about NEET Botany +1 chapter wise conceptual analysis and predicted weightage to different chapters. In today's lecture we will be discussing about NEET Botany +2. NEET Botany +2. In 12th class or +2 NEET Botany there are 4 different chapters, one, sexual reproduction in flowering plants. Sexual reproduction in flowering plants. Number 2, genetics. Number 3, microbes in human welfare. Number 4, ecology. Sexual reproduction in flowering plants approximate, that means predicted chapter weightage is around 9%. Whereas for genetics it is nearly 21%. Whereas for microbes in human welfare it is around 2%. Ecology is nearly around 19%. That means it's about predicted chapter wise NEET +2 botany. Okay, let us discuss one by one. Sexual reproduction in flowering plants, the first chapter in NEET botany. Before that we have to discuss and learn about, that means before going to the exact topic one has to learn about the basic aspects of reproduction. Reproduction. So two types, asexual and sexual. Yeah, in case of asexual, different asexual reproduction methods, for example, binary fission, budding, encystation, the gemmules, etc. No, at least we have to understand, at least we have to learn about different types and example. The gemmules in sponges, binary fish, for example, paramecium. Likewise, okay, budding in yeast. Okay, in case of plants, these are about microorganisms. In case of that means microorganisms and animals. In case of plants, we have to learn about different vegetative propagules. There are different vegetative propagules like runners, rhizome, offsets, suckers, bulbs, like that. Runners, for example, rhizomes, suckers, bulbs. Then it's all about different ways of asexual reproduction. In case of sexual reproduction, there are different stages. Broadly, you have to understand, because it's a complicated step, unlike asexual reproduction. Say it includes gametogenesis. That means with spermatogenesis, oogenesis, like that. Fertilization and embryogenesis. So we have to, under the reproduction, we have to learn about different things or aspects covers under asexual and sexual reproduction. Okay, then let us come to the main topic, that means sexual reproduction in flowering plants. Under the heading of sexual reproduction in flowering plants, it can be conveniently divided into 3 sub themes. Say for example, pre-fertilization events. That means structures and events. Then fertilization. Then post-fertilization. That means structures and events. In case of pre-fertilization events, in plants we have to learn about, say in case of male, stamens, microsporangium, pollen grains. In case of females, we have to learn about pistils, megasporangium, embryo sac. Okay. Stamen, I have discussed already. Structure and events. Say stamen consists of filament, anther. Then microsporangium. Say the different aspects of microsporangium. Say for example, tapetum layer that nourishes the growing, that gametes of male sex organs. Here, pollen grains. Say pollen grains, the outer layer, exine, intine, sporopollenin. Exine is made up of a chemical called sporopollenin. It's one of the most resistant chemicals so far, because no enzyme is discovered that can break down the sporopollenin. So we have to understand key aspects and events of different aspects of male sex organs. Microsporangium, like as I discussed, sporopollenin, for example, sporopollenin. In examination point of view, they might ask you in which layer does it present. It is present in exine, for example. Exine is outermost layer of pollen grain, likewise. Okay. Then stamen. Then with respect to female sex organ, that means gynoecium. Say pistils. It consists of stigma, style, and ovary. Just like here, the microsporangium, here the megasporangium and embryo sac. Different structures, that means description wise you have to understand and its important steps like this. Okay, it's about pre-fertilization, structure and events. With respect to fertilization, the most important aspect is double fertilization. Okay, in plants. Double fertilization is syngamy and triple fusion. Syngamy and triple fusion. Syngamy means one of the male gamete plus egg nucleus. Gives rise to zygote. Plus another male gamete. Another male, it combined with polar nuclei in the central cell. Nuclei, okay, polar nuclei. That leads to because one male gamete is haploid and there are two polar nuclei. That means two nuclei in the central cell. That means triple fusion. It leads to primary endosperm nucleus. Since two fertilizations are involved here, it is known as double fertilization. Zygote leads to embryo and primary endosperm nucleus leads to endosperm. That means you have to understand the events which leads to which. X leads to Y, Z leads to A, for example, like this. Here, triple fusion involves which aspects? Say the male gamete and the two polar nuclei present in the central cell. And it leads to, that means primary endosperm nucleus is a product of which? Triple fusion. And what exactly the double fertilization? Triple fission plus syngamy. And embryo comes from triple fission or syngamy. It's syngamy answer. Likewise, you have to analyze conceptually and study. Okay. Then with respect to post-fertilization structure and events, broadly you have to learn about seeds. That means different aspects of the embryos, embryo, endosperm, and seeds. Different things that covers under these things. That means under post-fertilization events. So these are the aspects of pre-fertilization and fertilization and post-fertilization. So different things covered under sexual reproduction in flowering plant. These are the major topics. Okay. Then regarding second unit, it's about genetics. Genetics and evolution. Now, evolution will be covered in zoology. In genetics, its predicted weightage is nearly 21%. Okay. Two sub-themes: principles of inheritance and variation. Then molecular basis of inheritance. Here, principles of inheritance means we have to learn about Mendel laws. We knew that Mendel has used 7 pairs of contrasting traits in pea plant, in garden pea. Say for example, seed shape. That means dominant trait and residual like that. Seed shape, round yellow. Seed color, yellow, green. Flower shape, that means flower color, violet and white. Pod shape, inflated, constricted. Pod color, green, yellow. Stem, tall, dwarf. And flower position, axial, terminal. 7 pairs of contrasting traits used by the Mendel to understand and to propose his observation, what we call Mendel laws. There are two kind based observations, based on monohybrid cross, monohybrid to dihybrid cross. Monohybrid cross leads to observation of first law, what we call law of dominance. Law of dominance. And another one is called law of segregation. Law of segregation. Dihybrid cross leads to law of independent assortment. Law of independent assortment. Simply monohybrid cross means cross between plants differing in one trait. So that's single trait. Dihybrid cross means differing in two traits. That means cross between plants differing in two traits. Say a law of dominance. Dominance means the factors occurs in pairs, they are discrete units, and in case of heterozygous condition, that means two different traits, one dominates the other. Is the law of dominance. Segregation, during gametogenesis, during gamete formation, each gamete receive one particular factor or allele. And in case of law of independent assortments, the two pairs of traits, wherein one pair of traits segregate from each other, that means independent to each other. One pair of trait independently segregate with that of another pair. Likewise, in just I am saying, say about monohybrid and dihybrid cross. Mostly you want to remember about the different ratios, Say for example 9:3:3:1. In case of dihybrid cross ratio, this is very, very important one. So that means round yellow, round green, likewise. You want to understand these things. Apart from Mendel, this is Mendel laws. Other aspects also have to cover, like chromosomal theory of inheritance. Chromosomal theory of inheritance, proposed by Sutton and Boveri. Sutton and Boveri. This is very important also. Okay, and sex determination under this chapter. Okay, sex determination. That means XXXO, ZO, ZZ, XX, XY, ZW, ZZ. Different ways of sex determination how to understand. For example, XXXY in human beings. How to understand different, that means type and organism. Example. Likewise, how to understand and learn. Then apart from this, other history aspects like disorders. I'm covering broad topics which we have to focus. Okay, disorders, genetic disorders, genetic disorders. There are two kinds, Mendelian disorders and chromosomal disorders. Chromosomal. Mendelian disorders particular to single gene. Okay. Chromosomal disorders with respect to absence or presence or alteration of chromosomes. These be aneuploid, polyploid, likewise. That means there are reasons I am saying. For example, during anaphase stage, if the chromosomes fail to segregate, either it may contain one extra or one option complete, likewise. That means non-disjunction. Okay, with respect to chromosome, I am saying. Say Mendelian disorders, with respect to single gene, say for instance, color blindness. Color blindness. Then it asks you, it is sex-linked residue disorder. Hemophilia. Hemophilia. Then sickle cell anemia. Sickle cell anemia. Sickle cell anemia is an autosomal residue disorder. Okay, residue trait. Then phenylketonuria. Phenylketonuria. It is because of resultant. It results because of absence of an enzyme that converts phenylalanine to tyrosine. Okay, phenylketonuria. Okay, thalassemia. Thalassemia. All these are the examples for Mendelian disorders. Chromosomal disorder, for example, Turner syndrome. Turner syndrome. Turner syndrome, for example, say trisomy 21. Turner syndrome, trisomy 21. Sorry, it's a Down syndrome. Okay, sorry. Down syndrome, trisomy 21. Then Klinefelter syndrome, XXY. That means in the 23rd pair of chromosome, one extra X chromosome. Okay, Turner syndrome, monosomy 21. Sorry, monosomy X, that 23rd, that means XO. The distinction between these two and these, that means Down syndrome, Klinefelter syndrome, with that of Turner syndrome is, in Down syndrome and Klinefelter syndrome, one chromosome is extra, that means additional chromosome. We got trisomy. Down syndrome trisomy 21. Klinefelter trisomy of 23rd pair, that means XXY. Now XY is a normal chromosomal complement, extra X chromosome. In case of Turner syndrome, XX is normal and one X chromosome is lost. That is monosomy. That's how we have to study. That means in examination point of view, they might ask you Down syndrome or the Klinefelter syndrome is resultant because of trisomy X or trisomy of 6 chromosomes or trisomy 21 or trisomy 16, like that. So we have to understand about these things and learn act accordingly. Okay, these are the different things we have to cover under genetic disorders. Apart from these things, other aspects like linkage and crossing over, we have to cover linkage and crossing over. That means under principles of inheritance and variation, these are the broad major topics that regulate that we have to much focus at much when compared to other topics. Okay, and with respect to the molecular base of inheritance, molecular basis of inheritance. Molecular basis of inheritance. So, as usually, we have to understand about double helical structure of DNA. Double helical structure of DNA by Watson and Crick model. Watson and Crick model of DNA. Okay, then DNA duplication. DNA replication or duplication. We have to study about semi-conservative model. Semi-conservative model. And Messelson and Stahl. Messelson and Stahl experimental verification, okay, of semi-conservative model. They proposed by Watson and Crick as well. Then apart from these, we have to learn about DNA packaging. DNA packaging. DNA packaging means histone concept, nucleosome concept. There are histone proteins in octamer of histone, H2A, H2B, H3, H4. These in 2 copies is octamer wherein 2 turns of DNA wraps around, because DNA is negative charged, histone proteins are positive charged, and they to form nucleosome, and nucleosome solenoid and long run chromatin. That means chromatin is nothing but a nucleoprotein. That means organization of DNA is in the form of chromatin. Beats on a string concept. We have to learn about this. Okay, DNA packaging system as well. We have to study. Then apart from these other topics, RNA old. That means we knew that RNA is the first genetic material. It is catalytic role. That means despite its genetic role, it also acts as a catalyst. Because since it is reactive, it is unstable. So DNA evolved from RNA because of its double standard nature and complementarity. That's why DNA is more stable product than RNA. That's why it is the best fit for genetic material. And under this head, we have to study about search for genetic material. Search for genetic material, like about Griffith transformation principle. Griffith transformation principle. If in the sense, say for example, streptococcus pneumonia, smooth strain, rough strain, there is certain thing that has been transferring from heat killed smooth strain to rough strain, that transferring the rough strain to smooth strain. That means a smooth strain means presence of a coat, a polysaccharide coat that give resistance to the strain, bacterial strain against the hydrolytic agents. So this is called transformation principle, and later onwards biochemical characterization done by Avery, McLeod and McCarthy, and finally Hershey and Chase. They stated that Hershey and Chase, they give unequivocal conclusion that DNA is a genetic material based on bacteriophage experiments. That's how we have to study about the steps for genetic material. Apart from these aspects, we learn about transcription. That means gene expression, transcription. That means mRNA formation. Then genetic code, translation. These are things we have to learn about. Genetic code, translation. Then gene regulation, That means gene regulation with respect to lac operon concept. Lac operon concept. Then human genome project HGP. Then DNA fingerprinting also we have to learn. DNA fingerprinting. So these are all about different aspects that one has to cover in molecular base of inheritance. Then genetics. After genetics, microbes in human welfare is very simple topic. It's also covered in zoology, but there is some slight question that may shuffle based on the closeness. Okay, say microbes used in household products, microbes used in industrial, microbes used in biogas, microbes as biofertilizers, microbes as biocontrol agents. Likewise, it has a meager weightage of nearly 2%. Okay, then the last important chapter and most important, ecology and environment. Ecology, that weighs nearly 19%. Under ecology there are different sub themes, organism and environment. Organism and environment, you have to learn about abiotic factors, abiotic factors like temperature, water, light, and different concepts like regulators. What are regulators? Regulators, conformers. Regulators means the animals like human beings, they used to maintain internal optic, the internal environment, what we call homeostasis. Say we have a constant body temperature, constant osmotic concentration, for example. That means these animals, that means for so-called regulators, they have the ability to maintain constant body temperature and constant osmotic concentration. What we call thermoregulation and osmoregulation. Say with respect to human being, thermoregulation. Despite the extreme environment, we have to maintain. Likewise, we have to understand conformers. Cope up, that means with the according to the external environment, these animals used to maintain likewise. Okay. Then we have to add different adaptations. Here, population we have to include. That means organism and how it respond to environment. For population, these are for convenience. I am saying, okay, for better understanding. Under population, we have to study about different characteristics. Characteristics like characters like natality, which affects the density of population. Mortality, immigration likewise, and then exponent growth models. Exponential growth, logistic growth model, different aspects. In population interaction, this is very important. In the examination point of view, they might ask you like description and example. Population interaction like mutualism, commensalism, amensalism, predation, parasitism. For instance, mutualism means both partners will get benefit. Commensalism means one partner will get benefit, other partner neither benefited nor affected. And amensalism means one partner will get affected and other partner will neither benefit nor affected. Predation, one partner will get benefit, other one will not. Other one will get effect. Parasitism, that means say host will get effect and parasite will get positive, will get benefit. Likewise, in competition, both will get affected. So these are different population interaction, different meanings, definition level, and with example to understand. Okay. Then the second aspect we have to cover under this ecology is ecosystem. Ecosystem structure and function. Is like productivity. We focus on ecosystem structure. The inputs means productivity. The energy flow, transfer of energy. How? And food chains, food web, nutrient cycle, and outputs. It's a decomposition, degradation. It's a broad overview, central dogma of ecosystem structure. And with the way you have to learn about productivity. Productivity means the rate of biomass production is productivity. For example, with respect to primary productivity and secondary productivity. Primary means gross primary productivity and net primary productivity. Gross primary productivity is overall production in the resultant because of a photosynthesis. GPP minus respiration leads to NPP. That's it in case of plants. It's up to producer level. Secondary production means CT, that means formation of organic, that means matter or by biomass at the consumer level. So productivity. Then decomposition, what to learn about decomposition, like fragmentation, leaching, catabolism, humidification, then mineralization. These are different steps involved in decomposition. Apart from these, to learn about different food chains, food chains. Different food chains. We have to learn. I'll discuss now how the energy is transferred. Food chains. food chains. Raising food chain, treated as food chain. Then food ecological pyramids, what to learn about. Ecological pyramids. Then nutrient cycles. Cycles. Say carbon cycle, phosphorus cycle, like this. So these are the different things that we have to learn in ecosystem. Okay. So these are the major topics. Apart from these, we have to learn about biodiversity and its conservation. Biodiversity and conservation. Learn about basics of biodiversity, genetic biodiversity, species biodiversity, ecological biodiversity, and losses of biodiversity and causes of biodiversity, causes of biodiversity, like habitat fragmentation, overexploitation, introduction of alien species. So these are all different causes for biodiversity. Then biodiversity conservation measures. In what way that we can conserve biodiversity? In situ and ex situ. In situ or on site, let's say national parks, biosphere reserves, sacred groups. So we have biodiversity hotspots across the globe. We have 34 biodiversity hotspots. In India, in Indian region, we have three biodiversity hotspots. Hotspot means where there is a highest species richness and endemism, and there is a threat for extinction. Threat of loss. So in ex situ means the geological parks, botanical gardens. To learn about biodiversity conservation measures. Then apart from these, they have to learn about other issues like in environmental aspect that we are going to learn. Environment, the last issue of ecology. In environment, say air pollution, say Air Prevention and Control of Pollution Act 1974. Likewise, 1981. Water Prevention and Control of Pollution 1974. Environment Protection Act 1986. So we have to understand about key aspects of that, means which affects the environment. Say pollution aspects. What I meant to say. Air pollution. Say under air pollution, so different case studies like in Delhi. Say to measures taken to contain the vehicular pollution. Say Bharat norms. That means in line with Euro norms. Say all these are mass emission controlling norms. Say the objective is to reduce the sulfur and aromatic content in the petrol and diesel. So to learn about that, okay, and role of compressed natural gas and how it is better than others. Then, in case of water pollution, then domestic sewage. I'm just covering the major topics, okay? Domestic sewage and industrial effluents. Under domestic sewage and industrial effluent, we knew that sewage means 0.1% impurities. So we have to learn about case studies like integrated wastewater treatment. This is very important. Okay. Then municipal sewage. So this is all about different things. Say sanitary fields. All we have to learn under this head. Then apart from these, agro-pollutants. That means pollutants arising from agriculture. Agro-effluents. Or toxic substances, for different words we can use for understanding I am using, okay. Then radioactive substances. Then most importantly, global warming and greenhouse effect. This is very important. Ozone depletion. That means Montreal Protocol. Say there are different summits like to conserve the World Summit on Sustainable Development 2002 at Johannesburg, Earth Summit 1991 at Rio de Janeiro, Montreal Protocol to contain respect to ozone depletion, and here conservation case studies like Chipko movement. We have to understand. Then joint forest management. In joint forest management, the people will get share for protecting the forest, and that means share in return. That means they have to protect the forest. In return, they will get some share, like fruits, gum, joint forest management. And Bishnoi tribe, how they protect the forest against kings. So we have to learn about the different things under conservation case studies. Okay. So under environment, pollution aspects, global warming, greenhouse effect. These are all description. Most of the part in ecology, major content of ecology is of description type. To to learn about different things, it's technical importance. Okay, so it's about different topics called on a NEET Botany +2. Okay, so respect to the different things, other aspects I have mentioned already. Revisions and taking mock tests is very important. Say NCERT books is main important source for NEET Botany and Zoology. Apart from these, I have careerorbit.com. It's an online source which I have referred to get knowledge about predicted weightage of NEET Botany and Zoology. Likewise, you can also refer certain standard and approximate, that means of your choice, to get more information, to get confidence which focus chapters are much important, and also appropriate references. So all this leads to that means all this has to in combination with regular revisions and taking mock tests. Okay, so that leads to holistic understanding. Okay, it's about NEET Botany. Thanks for watching.